Human Metapneumovirus, or hMPV, is a primary driver of acute lower respiratory tract infections. Structurally, it is very similar to its cousin, RSV, but with one critical difference. hMPV completely lacks the NS1 and NS2 non-structural proteins. Without those proteins, the virus induces a distinct inflammatory profile. Even after the initial fever breaks, patients often face a grueling post-viral phase, characterized by prolonged bronchiolitis and a lingering hacking cough. Clearing the virus does not automatically mean clearing the disease. Pulmonary recovery is a discrete, actively managed biological phase that requires entirely different cellular tools than the initial immune response. To repair the airway, the body must accomplish two distinct biochemical tasks. It must downregulate a specific inflammatory loop known as the hMGB1 RAGE axis, and it must restore the tissue's delicate redox balance. Standard antiviral suppression will not work here. Guiding a patient through this post-viral phase requires precise metabolic and nutritional modulation to switch the cellular machinery from defense to repair. hMPV evades the immune response by deploying G, P, and M22 proteins to block Rig-I and TLR7 signaling pathways. This silences the initial alarm, delaying the Th1 response and creating a pro-inflammatory environment flooded with cytokines like IL-6 and TNF-alpha. This inflammatory cascade triggers the release of a potent alarmin called hMGB1. Under normal conditions, hMGB1 stays safely inside the cell nucleus, stabilizing DNA. During infection, MLKL pushes hMGB1 out of the nucleus, causing an early release spike at the 6-hour mark. This hMGB1 binds to RAGE receptors on neighboring cells, creating a paracrine feed-forward loop that triggers a massive secondary release spike at 24 hours. Because RAGE is expressed at high levels in the lung, this feedback loop becomes self-sustaining. The receptors keep firing long after the viral load drops. The physiological toll is severe. The activated RAGE axis forces sustained NF-kappa B signaling, trapping the tissue in a chronic pro-inflammatory state. Simultaneously, it stimulates NADPH oxidase, flooding the area with reactive oxygen species. This oxidative stress directly damages pulmonary lipids, proteins, and DNA, leading to severe mitochondrial impairment across the lung tissue. The virus uses hMGB1 to ignite a biological fire that eventually burns on its own. It establishes a localized inflammatory environment that no longer requires active viral replication to cause extensive structural damage. This ongoing RAGE activation creates a distinct vulnerability to our diet. High-temperature cooking, like frying or grilling, generates dietary advanced glycation end products, or DAGs. When consumed, these glycotoxins do not simply pass through the digestive tract. Up to 30% of ingested DAGs are absorbed directly into systemic circulation. Comparing cooking methods, raw beef contains roughly 600 Ag units, but pan-frying drives it over 9,000. For a recovering patient, these dietary glycotoxins act as perfect exogenous ligands, crossing the lung-blood barrier to bind directly to sensitized RAGE receptors. The physical result of this secondary binding is a drastic impairment of alveolar capillary integrity, alongside a measurable increase in lung tissue resistance. This resistance effectively makes the lungs stiffer. The tissue loses its elasticity, making it significantly harder to ventilate and prolonging feelings of shortness of breath. Consuming high heat or fried foods during recovery actively supplies the body with external ligands. It feeds the exact inflammatory loop the immune system is trying to shut down, worsening structural damage. This RAGE-driven inflammation explains the persistent cough of hMPV. The airway epithelium is densely innervated by vagal sensory nerves, specifically A delta and C fibers. These neurons act as sensors for physical irritants. Under the constant stress of hMPV-induced inflammation, they undergo neuroplasticity, physically altering how they respond. When hMGB1 binds to RAGE on these sensory nerves, it impacts TRPRA1 and TRPV1 ion channels, sharply lowering their firing threshold. Because of this hypersensitivity, benign baseline stimuli generate full action potentials, triggering a severe cough without any physical irritant. Silencing this reflex requires direct interaction with these sensitized TRP receptors. a function that several natural botanical modulators are well suited to perform. Menthol activates the TRPM8 channel, which reliably inhibits surrounding respiratory reflexes. Compounds like gingerol and curcumin initially activate the TRPV1 and TRPA1 channels before forcing them into a refractory, desensitized state. The post-viral cough is a neuro-inflammatory reflex driven by the RAGE pathway. By utilizing specific botanical TRP modulators, we can directly blunt this nerve hypersensitivity. To resolve this pathology, clinical interventions must force a cellular pivot, shifting regulatory dominance away from the destructive RAGE axis and toward the reparative NRF2 pathway. When activated by compounds like sulforaphane, NRF2 translocates into the nucleus and binds to the antioxidant response element on the DNA. This triggers the release of protective enzymes like heme oxygenase 1 and glutathione, suppressing viral replication and inhibiting cell death. Once released, these enzymes significantly enhance the function of alveolar macrophages, boosting their ability to perform phagocytosis and rapidly clear out the accumulated viral debris. We can accelerate this clearance by supplementing with N-acetylcysteine, or NAC. NAC provides a targeted dual action. It acts as a mechanical mucolytic, and it serves as a direct molecular precursor for synthesizing more glutathione. By restoring glutathione levels, NAC directly neutralizes the oxidative stress generated by the RAGE axis. Simultaneously, it cleaves the dense disulfide bonds within the airways, mechanically thinning the trapped mucus. Adding a standardized extract like pycnogenol, derived from French maritime pine bark, compounds this effect. Pycnogenol chemically inhibits pro-inflammatory signaling while stimulating nitric oxide synthase to drastically enhance microcirculation. By upregulating the NRF2 pathway and providing precursors for glutathione synthesis, we biochemically antagonize the specific oxidative and inflammatory pathways initiated by the virus. Biochemical repair requires a parallel mechanical strategy. Prolonged sedentary bed rest during the recovery phase introduces severe physiological risks that can halt healing entirely. Without physical movement, respiratory cilia stop beating effectively. This causes thick mucus to stagnate, creating an ideal reservoir for secondary bacterial pneumonia. Clinical data points to a J-shaped relationship between physical exercise and infection recovery. Returning to moderate controlled activity restores the mechanical beating of cilia and strengthens the diaphragm to clear secretions. For patients struggling with deep tissue damage, targeted metabolic therapies like intermittent hypoxia-hyperoxia conditioning, or IHHC, offer an advanced method for driving tissue repair. Alternating between hypoxic and hyperoxic air triggers brief hypoxic dips. This releases HIF-1 alpha, a transcription factor that upregulates survival genes, commanding two primary physiological effects, forming new mitochondria and stimulating new blood vessel growth. The resulting burst of new blood vessel formation, combined with restored mitochondrial efficiency, provides the exact metabolic support required to repair the alveolar capillary barrier. Passive rest is only useful in the acute phase. To achieve full functional recovery, targeted mechanical movement and metabolic conditioning are essential to physically rebuild the respiratory architecture. Escaping an hMPV infection requires actively breaking the hMGB1 RAGE paracrine loop, fading on the left, and engaging NRF2 reparative pathways on the right. Introducing exogenous antagonists like dietary glycotoxins from fried foods or remaining sedentary keeps the tissue rigid. By strategically deploying targeted NRF2 activators and potent mucolytics like NAC, we suppress the localized oxidative stress and directly quiet the neuroinflammation driving the chronic cough. Pairing that biochemical strategy with a gradual return to physical mobility and advanced metabolic conditioning physically rebuilds the degraded pulmonary infrastructure. The post-viral phase is not a waiting period. Mitigating the severe, lingering symptoms of hMPV demands a systematic biological transition, shifting the body away from a passive state of viral defense and engineering an active state of cellular regeneration.